What is lysogenic conversion? Lysogenic conversion is the ability of some bacteriophages, also known as phages, which are basically viruses that can infiltrate and infect bacteria and reproduce within it. So lysogenic conversion is the ability of these bacteriophages to input their own DNA into a host bacterial cell chromosome and actually alter the phenotype of the bacterium, and in turn changing the efficiency of the bacterium. So this allows the bacteriophages to actually change what was once a harmless bacterium and into a disease-causing or pathogenic bacterium. It's one of many ways that phages are able to affect bacterial fitness and affect pathogenic bacterial evolution. So how is this different from the lytic cycle as far as replication? So in the lytic cycle, the phage attaches to the surface of the host bacterium through surface receptors and penetrates the bacterium through the cell wall, through the cell membrane, before moving on to the next stages of biosynthesis and maturation, basically creating new baby viruses, virions, that ultimately are released from the bacterial host cell through the final stage of release, where the host cell is lysed completely to release the baby viruses into the world. This is different from the lysogenic cycle because in the lytic cycle, the phage ends up killing the host through the lysing of the host cell to release the baby viruses, whereas in the lysogenic cycle, the host bacteria kind of acts as like an Airbnb kind of for the phage to rest and lay dormant. And because of this, the two cycles are also different because there's not progeny produced in the lysogenic cycle because the baby viruses are like never released through lysing the host cell like in the lytic cycle. And also the phenotype of the host bacterial cell it actually changes in the lysogenic cycle through the lysogenic conversion. So how is this related to pathogenicity? As far as pathogenicity goes, some lysogenic phages can actually carry genes that enhance the virulence of the bacterial host itself. These genes can eventually encourage the bacteria to release pathogenic toxins or have pathogenic effects, which traits that it didn't have before being infiltrated, basically. And also the lysogenic conversion allows for genetic recombination between the phages, which is contributing to the evolution of the phages and making them smarter and even more well-equipped to infect.